So now we have double-stranded DNA. It's stuck together and twisted around itself. The DNA has to be packed inside a tiny little microscopic cell. Remember, the human genome has 3 billion base pairs of DNA. That's a lot of DNA. And all that DNA has to take up a certain amount of physical space. We have to take that DNA and squash it down into a small structure so that it can fit inside a cell. In order for that to happen, there's a couple of accessory proteins that come into the picture, and that's what's shown here. There's something called chromatin. Chromatin is if you took a picture of the cell and you saw that there was a bunch of DNA mixed together with protein, you're going to call that chromatin. So chromatin is DNA plus protein. You can further break down the chromatin into nucleosomes. Nucleosomes is DNA that's also attached to protein, but a specific type of protein. And this specific type of protein is called an histone octamer. Shown here is an expanded view of one of these nucleosomes. You have histone proteins that are going to be bound by DNA. Now let's break down the actual structure of the histones. There's, as you imagine, it's an octamer, so there's 8 proteins. So there's going to be an H2A, an H2B, an H3, and an H4 histone protein. But there's going to be 2 copies. So 4 of these proteins, 2 copies of each gives you an octamer, or 8. Shown here is sort of half the octamer. On the other side of this, there would be 4 more proteins identical to these. There's also going to be sometimes an H1 histone associated. We'll talk about that momentarily. So for now, understand that a nucleosome is DNA that's bound by 8 proteins. Say 2 copies of H2A, 2 copies of H2B, 2 copies of H3, 2 copies of H4. Now, I'm going to tell you that this is an ionic association. So what charge does DNA have? Well, think of all the sugar phosphate backbones. It has a negative charge. The phosphate groups on the DNA are all negatively charged. So how are we going to get something to ionically associate with the negatively charged DNA? Well, we're going to give it a positive charge. So the histone proteins have a positive charge. The DNA has a negative charge. As you know, opposites attract, so they're going to stick together through an ionic association. Having said that, a simple point that the board exam wants you to remember is the amino acids that are abundant in histone proteins. Which amino acids are the positively charged amino acids? Well, it's actually there's 3 positively charged amino acids at physiologic pH carry a positive charge. There's histidine, arginine, and lysine. So histidine, arginine, and lysine. And of those 3 basic amino acids, two are abundant in histone proteins. And which are they? Well, it would be nice if the histidine was present in histones, but it's not, because biochemists don't make sense, right? No, it's going to be arginine and lysine. You need to remember that. Arginine and lysine are abundant in histone proteins. That's what gives the histones their negative charge. The histones are positive charge. DNA, their negative charge, allows them to associate with each other. Incidentally, what's a pirate's favorite amino acid? It's arginine. Okay. So, 2 types of chromatin. When we talk about chromatin, we have heterochromatin, which would be dark staining areas in an electron micrograph. We have euchromatin, which are light staining areas in an electron micrograph that I'll show you here in a minute. Now, what's the difference between heterochromatin and euchromatin? In simple terms, heterochromatin is areas of the DNA where there's inactive genes. So that means that there's low amounts of gene expression occurring. We're going to find in the X chromosome that's been inactivated in each of a female's body, the cells of her body, there's a bar body. So for the most part, the bar body would be found within heterochromatin. What about euchromatin? Well, euchromatin is going to be areas of active genes. It's going to be areas where the cell may be undergoing apoptosis. So a classic question is to maybe give you an electron micrograph, point to a lighter staining or a darker staining area of the cell, and then ask what's going on in this particular portion of the cell. If it's light staining, you need to recognize that as euchromatin. Perhaps there's active gene expression going on there. Perhaps the cell's undergoing apoptosis. If there's diffuse euchromatin present. Whereas if it's all dark staining areas, that would be areas of inactive gene expression, which is mostly going to be in the form of heterochromatin. You can sort of picture it this way. When the DNA is inactive, it's going to be tightly packed together. And so when you do an electron micrograph, the electrons can't penetrate through that tightly packed DNA material. So what ends up happening is when the film is developed, it looks very dark. If the gene expression is active, the DNA is going to be relaxed and separated. The electrons will be able to readily pass through the DNA at that particular area, and it'll end up as a lighter staining area on the photograph. So, looking down here now at our structure, you'll see that we have either a 30 nanometer or a 10 nanometer particle. The 10 nanometer particle, when you read this, what's being referred to is simply the nucleosome, the core octamer, that's being encompassed twice by the DNA. So that's the 10 nanometer particle. If enough 10 nanometer particles come together, they can form a regular arrangement, which is called the 30 nanometer particle, shown over here. What I want you to appreciate from this is that the 30 nanometer particle is much more tightly associated than the 10 nanometer particle. And there's one other histone that we have to discuss that actually helps hold the DNA into the 30 nanometer particle. And you guessed it, that's the one shown here in blue. It's the H1 histone. The H1 histone, I like to think of this sort of as the general. It's the general, and here's all the troops. The troops are these core nucleosome octamers wrapped up with DNA. When the H1's histone is there, the troops are all lined up at attention, and they're tightly packed shoulder to shoulder. And actually, this helps keep the DNA protected from degradation. So the analogy applies. When the H1 histone is there, the DNA is going to be present in the 30 nanometer regular arrangement, and it's going to be resistant to nuclease activity. When the H1 is absent, without H1, without the general, when the cat's away, the mice will play, right? So when the H1 histone leaves, there's going to be openings in the DNA that could be cleaved by nuclease activity. So, 30 nanometer particle, resistant to DNA cleavage. 10 nanometer particle, more sensitive to nuclease cleavage. A key point to keep in mind. Here's showing you a figure that just compares the various structures of DNA as you start to make it into a higher order structured material. Shown at the simplest level is the DNA double helix. Then that will, remember, associate with a nucleosome protein. You have the H2A, H2B, H3, and H4. 2 copies of each of those is going to associate with each of these. It's what gives you this beads on a string appearance to the nucleosome. That's the 10. Each of these is a 10 nanometer particle. Enough of these associate, and along comes the H1 histone. Now we have a 30 nanometer chromatin particle. Enough of these 30 nanometer particles associate, well, now you form these rosettes and these loops, and you have additional scaffolding proteins which help hold the DNA in a higher ordered structure. And finally, these can associate with other rosettes, and you get a very highly ordered packaging of DNA. So you take all of this material, you squeeze it into a small space, and it can fit inside the cell. The question now becomes, which of these various forms of DNA is heterochromatin, which is euchromatin? Now, in truth, all of these forms of DNA, the double helix, the 10 nanometer, the 30 nanometer, even the rosettes, you can find euchromatin in these ordered structures. If you find the higher order packaging, like up here, it's probably going to be heterochromatin. Now, not everything is black or white like that. In reality, you should think of it this way. The higher the packaging that goes on, i.e. the more it goes from double helix all the way up to these association of these rosettes in this higher order packaging, the less transcriptional activity is going to be going on. The more it's, I should say it this way, the less highly ordered the structure is, the more transcriptionally active it will be. So if you're just visualizing DNA at the double helix or even the 10 nanometer particle form, maybe even the 30 nanometer particle form, this is going to be more transcriptionally active. Whereas if you look at a DNA that's very tightly packed, i.e. it's heterochromatin, it's going to be less transcriptionally active. Here's an electron micrograph that's actually showing you what the cell, the nucleus within the cell looks like. So shown here is the nucleus of a eukaryotic cell, obviously. You can even see some of the endoplasmic reticulum in here, perhaps some other organelles within the cell. But within the nucleus, you could see that it's subdivided into smaller structures. In particular, you have the nucleolus within the confines of the nucleus. It's important you remember what that structure does. Do you remember? Well, the nucleolus is where you get assembly of ribosomal RNA. So the ribosomal RNA gets synthesized and assembled in the nucleolus prior to being released out into the cytoplasm. Now, if you look at where this arrow is pointing to, you see a lighter staining area of the cell. That's going to tend to be euchromatin. Remember, euchromatin is transcriptionally active. The DNA is going to be less tightly packaged, so the light's going to pass through, which will look like a lighter spot on this electron micrograph. Compare that with what you see at the edge of this arrow for heterochromatin, this darker staining area. You see some more up here, here, some more over here. The heterochromatin is the tightly packed areas where the DNA is transcriptionally inactive. The light and electrons can't penetrate through that area, so it ends up as a darker staining area on the electron micrograph. What biochemical events happen in order to cause DNA to either be heterochromatin or euchromatin? This is an area where they like to test you. So let's look first at DNA methylation, probably the most important step. DNA methylation is going to inactivate transcription. So when DNA becomes methylated, what's going to happen is it's going to change its structure. And it actually causes the DNA to become perhaps more tightly packaged and less likely for it to be expressed in the form of transcription and translation. So another way of saying that is DNA methylation favors heterochromatin. It favors heterochromatin or inactive transcription because you won't get good gene expression going on in any gene that's methylated. The next step down here is histone acetylation or phosphorylation. Histone acetylation and phosphorylation actually works the opposite. When histones become acetylated or they become phosphorylated, what they end up doing is losing some of the positive charge that holds them in contact with the DNA. If they lose the positive charge, they're going to dissociate from the DNA, and now the DNA is more accessible for enzymes to come in and start transcribing, and then later translating the RNA into protein. So when you see, if you read a case or some sort of experiment that's been done, and something's been done to alter the acetylation phosphorylation status of the DNA, or pardon me, of the histones, if the histones have been more acetylated or phosphorylated, you're going to see more active transcription because the positive charge has been lessened and the interaction of the histone with the DNA is less. So you can either modify the DNA through methylation. Methylation inactivates transcription. Or you can modify the histones. Histone phosphorylation or histone acetylation is going to favor gene expression. That brings us to the close of Chapter 1, in which we discussed nucleic acid structure and organization. As I mentioned, the actual structures of the nucleic acids not too important for you to try to memorize every bond in one of those structures. It's more important for you to understand the differences in the structures between the purines, between the pyrimidines, understanding the differences in structure between RNA and DNA, and then understanding how DNA gets packaged into higher order molecules so that it can fit within a tiny little cell.